Hi ladies, just a quick little revision mind map on osmosis. So one of the topics that we do in fifth year that some people kind of struggle with that I don't think is too difficult once you learn your definitions and your key experiment off. So let's start off with first of all our definition of osmosis, okay? So it's the movement of water molecules from areas of high concentration to low concentration, and then the important bit, across a semi-permeable membrane. We'll define that now in a second. Okay. So a semi-permeable membrane then, girls, okay? So semi-permeable membrane is one which allows certain materials to go through, okay? So here we've got, this is our membrane here, all right? And these are different particles that are on one side of it, all right? So it's semi-permeable because it only allows these small particles to move back and forth across the membrane. So essentially there's tiny little holes or pores in the membrane, so it only allows the small particles to go through. It won't allow these larger particles to go through. Now examples of semi-permeable membranes in our body, girls, will be your cell membrane, okay, will be other semi-permeable membranes, viscing tubing, we'll talk about that in a second. Okay, that's something that we use to model the cell membrane during our experiment, our key experiment, which we'll talk about down here in a second. And we also need to talk about diffusion. Okay, so diffusion is essentially the movement of particles from an area of high concentration to low concentration, but it can be anything. It doesn't have to be water. It doesn't have to be across a semi-permeable membrane. Okay, now so diffusion, so movement of particles across movement of particles from areas of high concentration to low concentration. Sorry, that's a bit squished there, I know. Okay, so movement of particles from areas of high concentration to areas of low concentration. So apologies now, that is it's not the best there, okay? Now, in terms of diffusion, girls, examples in our body of diffusion is O2 in the lungs. Okay, also we see it in kidneys. Okay, we see diffusion of certain substances in the nephron. We also see what's called active transport, okay? I'll just write that down here. So active transport is the movement of materials using pumps and ATP. Okay, active transport is the opposite of osmosis and diffusion. Osmosis and diffusion are what are called passive transport. They do not require any energy to happen, okay? Whereas active transport does require ATP to occur, and it usually only occurs for larger molecules. Okay, such as sugars, such as proteins, such as enzymes. That's where you're going to see at most active transport, okay? So just always remember, in the body, you've got two types of transport systems. You've got passive transport, you've got active transport. Passive, no energy. Diffusion, osmosis. Osmosis is only movement of water. So again, we see that in the kidneys and the nephron. You also see it in plants and their roots, okay? So importance of osmosis is plants. Pen is very dodgy, apologies. Okay, so plants in the roots, Okay, you also see it you also see it in cells in animals, also in the kidneys. Okay, in the nephron. All right. So they're the importance of osmosis. Okay. Remember, in plants, girls, it's osmosis that encourages water to move up the plant. So remember, we talked about your tension cohesion model. So osmotic pressure was one of the parts of that, and also it's what keeps plants what's called turgid. Okay, turgid means that they have enough water in their cells to remain upright and standing straight, so they can get enough sunlight to be able to do photosynthesis. Okay. Now the hard thing, or the thing that comes up most often, girls, for osmosis is your key experiment. So your key experiment looks like the following. So we've got 2 beakers. With your visking tubings, which essentially look like little sausages or little sweets. Okay. Okay. One visking tubing is your control, which is water. Okay, so visking tubing. So remember, your visking tubing, like I've said, is what is modelling the cell membrane, okay? So it's a semi-permeable membrane. And then we have your water, which is your control, and this is obviously water as well. So we shouldn't see, when we pre-weigh this visking tubing, we shouldn't see a dramatic increase or decrease in weight, okay? This one, girls, contains sugar plus water. Okay, and we should see an increase in mass of this visking tubing over time because osmosis. So what we get, girls, I'll just put little arrows in here if my pen will work. So your the water will enter the visking tubing through your visking tubing there. Okay, so we'll see osmosis in your sugar sample. We won't see any osmosis, or we should see very little osmosis in your control sample. So this one will become larger over time, and we should see an increase in mass. This one won't become larger over time. It should kind of stay very similar size. Okay, now key terms. Right, so your key terms, girls, for your osmosis, I haven't left myself a huge amount of room there, so apologies. Hopefully I'll be able to fit these in. Okay. So key terms. So key terms, girls, I've got a new pen. Hopefully this is working a little bit better. So your key terms are the following. So what you need to know. So you need to know the terms hypertonic, hypotonic, and the hypertonic, hypotonic, and isotonic. There's 1 or 2 more as well there. So let's write down the definitions for these guys. So hypertonic is where it's the cell is in a very concentrated solution of sugar or salt, okay? Now, when a cell is in a very concentrated solution of sugar or salt, what happens, girls, is it loses water and it what's called crenates. Okay, it shrivels up. I think I've spelled that wrong. Okay, so crenates. Hypotonic, girls, that's where a cell is in a very dilute solution. Okay, and what happens here, girls? It does the opposite of crenation. It swells up and it undergoes what's called lysis, which means it bursts. Okay, so swells and bursts, which would be a very bad thing, obviously. Okay, and then isotonic is the cell's cytoplasm is the same concentration as the surrounding media. Okay, so the cell remains perfect. And essentially, girls, this is what your blood should be. Your blood should always be isotonic to your cytoplasm to prevent your cells from swelling and undergoing lysis, or from shriveling and undergoing crenation. Now, when they talk about crenation in your textbooks, girls, they usually show a picture of a red blood cell, and the red blood cell essentially looks like a raisin. And this doesn't happen for plant cells. The reason being, and also this doesn't happen, lysis doesn't happen for plant cells either, because plant cells are protected by the fact they've got a rigid cell wall made of cellulose. So what happens to plant cells, girls, is that they undergo what's called plasmolysis, where the cytoplasm comes away slightly from the cell, from the cell membrane comes away slightly from the cell wall. So the plant becomes wilted. Okay, so it essentially falls over. The cells aren't what's called turgid, like we talked about already. So plant cells are slightly different because they do have that rigid cell wall, and like they don't undergo lysis either. They can't burst. So that means that they can remain, they can remain in one piece when they take in too much water, and actually they will always have slightly too much water in them because it allows them to remain rigid and it gives them a little bit more support, because remember, plants don't have bones or anything like that. Okay, so that is mostly osmosis, girls. Apologies now for the dodgy pen. Hopefully that is useful.